Bienvenidos todos, welcome all. I'm Mike Wong, a postdoc in astronomy and astrobiology, and it is my pleasure to introduce our speaker today, Abel Méndez. But before we get onto that, I would like to begin with this brief land acknowledgement. So we acknowledge that the UW Astrobiology program resides on the land of the Coast Salish peoples, land that touches the shared waters of all tribes and bands within the Duwamish, Puyallup, Suquamish, Tulalip, and Muckleshoot nations. And we recognize that we, the UW Astrobiology community, benefit from the occupation and use of this land, and that it is our responsibility to mend and forge our relationship with Indigenous peoples who are extant and vibrant members of the greater Seattle community. And I'll put in the chat some helpful links to resources if you would like to learn more. Okay, and with that, it is my great pleasure to introduce Professor Abel Méndez from the University of Puerto Rico at Arecibo. Dr. Mendez received his PhD in physics and biophysics from the University of Puerto Rico at Mayagüez. He is now the director of the Planetary Habitability Laboratory at UPR Arecibo. He and his group are best known for developing the Earth Similarity Index, the Visible Paleo-Earth Project, and maintaining the Habitable Exoplanets catalog. Now, anyone who has sold their soul to the blue bird knows that it's a good day on Twitter whenever you get retweeted by Abel. His substantial Twitter following of scientists and lay people alike means that in addition to being a cutting-edge researcher, Abel is an important hub for facilitating the communication of all of our research and a powerful force for promoting diversity in academia. So without further ado, please join me in welcoming Professor Abel Méndez as he tells us about his latest work on quantifying planetary habitability. Abel, the floor is yours.
Thank you, Mike. Thank you for that introduction. I am Professor Abel Méndez from the Planetary Habitability Laboratory at the University of Puerto Rico at Arecibo, and I will be talking about habitability models in astrobiology. These models are developing, we are developing this model with many colleagues to study the potential for life in the universe from microenvironments to global biospheres. Planetary habitability is an old problem. In 1883, William Miller pondered that the habitability of stellar planets, an old name for exoplanets, could only be determined relative to Earth. We know many of the planetary and stellar factors driving planetary habitability. The problem is how to convert them to a measured habitability. So this is the big problem right now of the astrobiology community, how to connect so many variables, how to make sense of them. And that's what I will be talking today. Common assumptions about habitability. First, that we can not define habitability, but I will show you that this is a well-defined concept in biology since at least the last 40 years. That it is impossible to quantify habitability because we might not have enough information about environments, but you don't need all the information to assess habitability because it is measured by parts. Finally, that any habitability model pretends to determine if places are habitable or not, which we know this is an impossible task. We all understand that this is our wish, but this is not the main purpose of habitability models. Let me explain, giving an example. Here are 6 types of environments. We know that the terrestrial ones are habitable, and we presume that the planetary ones here, Mars, the Moon, and Venus, are not. The only way to determine if any of these places are habitable in the common sense is by finding or putting life on them, which is observable. Life is observable in the terrestrial case, at least in the dry forest and the rainforest. So seeing habitability models are not life detection instruments, although they might provide some guide. The purpose instead is to understand why the difference, why some places are better, but also why some places are worse. In other words, these habitability models help to identify and understand the key environmental variables or limiting factor controlling the abundance of life. So the binary habitable, not habitable solution is an oversimplification of what habitability models do, leading to misinterpretation of their application. So here's a very general definition, some general definition of habitability. I started asking myself about how to define and measure habitability around 20 years ago in the early days of the astrobiology field. I started by looking for a definition, including beyond the astrobiology field. Most dictionary definitions were associated habitability with more human habitation. This topic seemed very important, deserving a whole chapter in any biology textbook. And I went to many textbooks and many papers not related to astrobiology, looking for the words. And to my surprise, the words habitable and habitability were not even there in biology textbooks. And for me, that was something rare. So therefore, I thought, like everybody else, that the topic of habitability was a new problem or question in biology brought by the astrobiology field, that the biological concept of habitability was exclusively an astrobiology topic. The first astrobiology paper to directly address how to quantify habitability was published by Chuck and Holland in Astrobiology in 2007. They suggested using available power for life or energy per time as the best proxy to quantify habitability. A paper around that time by Tori Holler also agreed with this definition. I was fascinated with this idea, but not totally convinced it was the final solution. Then around 2009, I found something very interesting. I found a 1993 paper by Rice and others, not related to astrobiology, but using the word habitability to refer to the quality of a semi-arid land for some types of birds. The paper also pointed that their model was consistent with the Habitat Suitability Index used in ecology. And this was the first time I see this phrase. I searched for habitat suitability and went to the search, and I was overblown. I finally realized that what astrobiologists call habitability as a new problem was actually an old problem called habitat suitability in biology. In the late 1970s, ecologists were using different measures of habitat quality, not all consistent between them. Same problem astrobiologists are facing today. Then in the 1980s, the US Fish and Wildlife Service decided to fix this and create a standard described in their white paper, Habitat Suitability Index Models. So this was an amazing resource. And I then decided to look back on all this knowledge for the last years. So let's formalize some basic concepts of habitability, or more formally, habitat suitability, but I will use the word habitability because people are more used to that word, at least in this context. Habitability is defined as the suitability of an environment for life. It's proportional to the carrying capacity. And this is the key here, how to measure habitability. This carrying capacity is the maximum stable biomass that an environment can sustain. This is exactly how habitability is quantified in practice. Finally, habitability is used to determine how a set of environmental factors impact life, their presence of abundance. The key thing here about this statement is that the set of environmental factors are not necessarily the only important ones. So you don't need to know all the environmental factors to measure habitability. You only you're selecting some variables because you want to understand how significant or not are those particular variables for life. Here is the simplified general equation used to quantify habitability in biology. The study conditions are based on a set of variables of interest compared to some standards, as long as they are proportional to the carrying capacity of the system under study. In practice, you construct a library of models. It's not that you have this equation and master and create just an equation for each case, but from this equation is a starting point to create a library of models describing one particular scenario, each for the specific environment and factor of interest. And I love this analogy. It simplifies things very well. Here is a powerful analogy of the requirement of life that is useful to understand the limit of life and habitability. Life needs the classical elements: air, water, earth, and fire. Or more correctly, a gas, a liquid, solid, and some energy. One of the reasons is that not all elements for DNA are available in a single phase of matter. So you need them to be mixed together with a source of energy. It is easier now to understand the abundance of life in terrestrial environments using the classical elements analogy. So in a rainforest, you have all these ingredients in abundance, and that's why life is so present here and visible, because macro life, vegetation, animal life need a lot of these resources. So forests can sustain a large biomass. But if we consider deserts, ice cap, clouds, and the surface or deep ocean, they have strong limitations of one or more of these elements. Thus, although they are habitable environments, all these samples are habitable environments, the amount of biomass per unit of space and time never compares to that of forests. The classical element analogy is a good qualitative tool, but we can do much better to quantify habitability than this. And here is a model that we are working on. It's an extension of the habitability models using ecologies where habitability is proportional to the mass and energy available for life. Only a small fraction of the total mass and energy of a system is available for life. Here in the red box is the differential expression where we can see that habitability depends on fluxes of mass and energy. And we can explore with this equation equilibrium condition. It is always necessary to define a region of interest in space and time, like I'm showing here, whenever you want to try to assess the habitability of a system. That could be a patch of soil, a volume of water or air, or even a planetary surface, if you want to consider the habitability of a large environment. Here in the blue box is a very convenient way to calculate habitability relative to some other known environment and normalize in a scale between 0 to 1. So that's what biologists usually do, try to always calculate habitability relative to some environment of interest for comparison purposes. This also helped to cancel out the quality factor. So here I said that it depends on the mass and energy, but there's always a quality factor. And most of the biology is concentrated in this quality factor. And you don't know how fraction of the mass and energy is used by a particular life. But if you are comparing with other environments, and you're assuming that they share the same fraction, the fraction cancel out, and it makes much easier to combine things. We can use the analogy of an electric circuit here to the right to understand how to combine many parameters in the equations. All necessary substrates like nutrients, for example, from the gas, liquid, or solid phase are used in parallel, and thus can be aggregate. And this is a key word here in the ecology models. How to aggregate, how to combine the different environmental variables. So here they can be aggregated as resistors in parallel. Then the total mass required for life becomes a reduced mass, constrained by the limited substrate from all available substrates. In our model, the first step is to pick the variables of interest and calculate the environmental fluxes to these variables. So you start with deciding what variables, these variables, you know already that they're important for habitability. Maybe some you don't know if they are that important. Some are because they are easy to measure. And from that, you calculate how these variables that you pick, you have to calculate fluxes of mass and energy. So here is some physics and chemistry to do, depending on, you can divide it, and that's why the classical elements analogy is help to organize them. So you decide then what will be the particular fluxes that you will be interested in depending on those variables. Then apply an ecophysiology response curve, which will be the Q factor in the equation, which is the hardest part to do because most of the biology is here in this part of the calculation. This response curves includes most of the biology. And let me give you a more detailed sample. This is a sample of the general model of an ecophysiology response curve that can be used to describe the effect of many state variables on life, from microorganisms to plants and animals. Life is usually constrained by some limit for each variable of interest, temperature, pressure, and has an optimum at some value between these limits. In plot 8 here, we are using this general model with a shape factor equal to 1 that has been successfully used to describe the growth rate of microbial life as a function of pH, water activity, or even temperature in plants. In plot B, a model for S equal 2 has been successful to describe the growth rate of microbial life as a function of temperature. A library of these models can be used to construct a new response curve for a new variable. Then we can go back and apply these models to our previous example of terrestrial environments, but now quantify with the idea of the classical elements. And this is in particular with respect to the available oxygen, water, nutrients, and temperature. We get, and using the forest as the standard of comparison, that forests are the most habitable place in our planet. That is where most of the mass and energy resources are available for life, while clouds in the other side of the spectrum are the worst habitable environment in our planet. All these places are habitable, but at least I can tell you, depending on the variables that I pick, how good are or how we can compare them in terms of this mass energy habitability. Then we can go further and let's try to apply this model to other interesting cases. Let's see how to construct the habitability model applied to the surface of Mars as an example. The first step is to select a space and type of interest. So here we are taking a crater. We then select some variables of interest here, surface temperature and relative humidity, because we know these two quantities, how they impact life, but also are available from Mars Curiosity for Gate Crater. We then select in step 3 microbial life as our life of interest. So any comparison will be, for example, here endolytic life from the Atacama Desert. And at this point, we could look for any known ecophysiology response curve of any of our variables of interest, temperature and relative. And there are, for temperature and relative humidity, we have different response curves that we can use. In step 4, we pick a standard of comparison. Now the place of comparison, not the life of comparison, but the place of comparison that Atacama Desert in Chile. This means that we are calculating habitability relative to this terrestrial environment, an environment that we know. We then parameterize our variables into our mass energy model. The key here is to find how your variables relate to the mass and energy and quality of the environment. Usually this is not hard to do. The Q parameter encloses information about the quality of fraction of the environment available for life. So in this Q parameter is the biology here. And that's why we have two terms, one related to the response to temperature and one related to response of relative humidity. Finally, we try to validate, if possible, with our standard of comparison. So this is an optional step, but if you are proposing a habitability model, make sure it works for Earth first. So this model could be applied to other terrestrial environments as to see if there is proportion between this habitability and biomass. We apply the model to all possible temperatures and relative humidity variation on Earth and Mars. The special region here show conditions necessary for life as we know it, temperatures over -18 degrees Celsius and water activities about 0.6. The surface of Mars never touched these regions and must stay temperatures and relative humidity still low here, close to where this equation is. And this is the actual equation of the model that I use here. But from the model, we can see how bad it gets on Mars. So here in the scale, 1 is relative to our Atacama Desert. And we see that the surface of Mars is thousands of times less habitable than Earth, even considering just temperature and available water. This is excluding other important factors. And this is something good to know, that not considering other factors that might be make things worse is already worse because of only these two factors. Let's go straight to more details. Looking at Mars in more detail in the bottom plots here. Here is a thermal humidity habitability for the two different Mars sols, a cave crater, using data from Curiosity. Here, the top are temperature and relative humidity during the one sol, but here is the calculation of habitability. These temperatures and humidities are too low for life as we know it, so we are not including the response curve, because if we include the response curve here in the problem, then all habitability will go to 0. And we don't want to make any bad environment 0 because we won't be able to compare it. So we are not taking in this calculation the response curve. We notice that in both hot and wet sols, the total habitable time per day is around the same, about 9 hours, and peak close to 3 to 4 hours after noon. Nevertheless, upper habitability values are over 1,000 times less than anything in the Atacama Desert. So even if there is a life form which has a response curve that says that they can tolerate this low temperature and low humidities, it will be life as we don't know it. Because no matter what life you're putting here, it has over 1,000 times less water and energy resources. And no other factor will make that to change. So at least you know how bad it is Mars compared to the Atacama Desert. In other words, this is a polite way to say non-habitable. Then let's go further, how to construct a habitability model for exoplanets. Suppose we start to learn more about exoplanets, like the size of the atmosphere and water content, something that we might do in the next decades. Then we might be able to compare these planets in terms of the potential for life. Let's see how to construct a habitability model for a global biosphere as an example. The first step, as before, is to select a sample of interest. Now my sample is the near surface layer, because I want to evaluate the global biosphere. We select some variables of interest. So the variables of interest, because are easier and are global properties, are planet radius. We know, and we know this for many exoplanets, but we don't know the surface temperature, less the surface pressure or the ocean fracture. But at least we can explore the possibilities, especially if we are trying to do general models. We then select our life of interest will be for primary producers on land, because they are the base for other life forms. We pick a standard comparison, the same surface, but for Earth, and the same properties. Here again, the same, you have the same problem of the quality, and you try to select the variables, make it proportional to how these variables relate to the mass available in this layer, to the energy available in this layer, in this case from light from the star, and the quality factor, which depends on the temperature on what primary producers require. Finally, we try to validate our model. We don't have the luxury to validate our model with any other global biosphere in the universe. Earth is the only biosphere as an example. But as we did with Earth, by creating a 2D version of our model that correlates with the carrying capacity. So what we did, found a linear relationship between this quantity calculated in the same way, but for the whole planet in terms of for the different terrestrial biomes. And we found that those biomes are, this habitability for different biomes is proportional to the net primary productivity, which is the biomass produced by unit space and time. This means that, so that's one way to validate your model. This means that our global biosphere habitability model can be used to constrain the potential total biomass relative to Earth that can be produced in an Earth-like planet, but only as a function of our variable of interest. Like everything else was okay, and these were the only conditions necessary for life. Temperature, as you saw, temperature is an important factor in this model, starting with that. Not only surface pressure, but also ocean fraction. But starting with temperature, so we are taking special attention on how to model temperature. In 2017, we explored the feral eccentricity and discovered that the mean global equilibrium temperature of planets decrease with eccentricity, contrary to what is initially expected. The flux do otherwise. So flux increase with eccentricity, which is the red line here, but to our surprise, the mean global equilibrium temperature decrease, which is the blue line here, with eccentricity. So if we, and this model we validate with the solar system, Mars, and some simulations. And here are the analytic expression. So we were able to calculate analytic expression. But the usual thing is that we believe that while calculating these equilibrium temperatures, it was fine to use the average flux and put in the equilibrium temperature equation, but this is totally incorrect. That will give you values much higher than for real. We are focusing now on surface temperature, and we discuss in this same paper how to calculate the mean global surface temperature, now moving from the equilibrium to surface temperature, giving a seldom used parameter called the normalized greenhouse. So we have this analytic expression that we know can be used to predict the surface temperature on a planet. Here is the albedo, the bond albedo. But now we have this normalized greenhouse effect, which is a number between 0 and 1, very convenient. As the bond albedo, this is another important number that summarizes the effect of an atmosphere on the thermal response of the planet. So you need two numbers, the albedo and the normalized greenhouse. Here's this table shows these values, including one additional one for the solar system object. This kappa, we call this kappa the surface thermal factor, which combine both the effect of the albedo and greenhouse. So all this expression involving the albedo and the greenhouse and the 1/4 of the power is this kappa here. And we found an interesting relation that we noticed for the solar system and simulation that kappa is about 1 for planets with none to 1 bar atmosphere. And it does not depend much in the stellar flux, but somehow, yes, in the spectrum of the star and the composition of the atmosphere. But kappa strongly depends on the surface pressure of the planet. And here is the plot for the simulations we have done. And this the blue line is for G stars and this from M star. So kappa is equal to some constant which depends on the stellar flux and in the stellar spectrum and the properties of the atmosphere, but is strongly dependent on the square root of the surface pressure. And this is a very convenient equation because then something interesting here, we got that therefore surface temperatures are easier to predict than equilibrium temperatures, given that you know the atmosphere surface pressure. At least that makes much easier things. And again, here I'm showing all the models that we try, including published models like Rocket 3D, but some runs that we did from different sources. At least we try 40 different types of models, 1D from 3D. And solar system objects are in pink here, and you see Titan, Umbriel, up to Pluto here, Earth and Mars right here. This is kappa equal to 1. And most of the simulation concentrate here, most of the simulations from the Rocket 3D, for example, which must include atmospheres similar in, at least in pressure to Earth, are concentrated in this region. But all these points here correspond to high thick atmosphere, 10 bars or more. And as you can see, they correspond for different typical values. So for kappa equal 2, you see that those that simulate almost the surface temperature of Venus, then kappa equal 2 for all the flux change. So even from fluxes equal to Earth value to up to 10 values, kappa is about the same, just the same value. So this this type of model provides a simple analytical expression to constrain the surface temperature. And here is for 10 bars CO2 atmosphere, this simple expression that you can use with to constrain what will be the expected temperatures for these atmospheres, at least of CO2. So the zero albedo equilibrium temperature of an exoplanet, which is this part here, is similar to the surface temperature of rocky worlds with 15 atmospheres. Here in red, we constrain the surface temperature of the 7 planets in the TRAPPIST system, assuming albedos and greenhouse for none to thick atmospheres. And I have to stress out that the interesting thing of this kappa number is that if the planet gets closer, the albedo will change and the greenhouse will change. But this fraction, which gives kappa, it will stay about the same. So that's important point. Here in green are upper habitability limits using our global habitability model. Only planets D and E have a non-zero value, so they are shown here. We E slightly better, but both lower than Earth. So how we can interpret analysis like this? For example, we could say that any biosignature signal, if any, is more likely to be stronger in planet E than in D. If we find the reverse is true, then this might mean, if the observations are correct, this might mean that the different biology process is at work, something not like Earth as expected, since our model is based on terrestrial biology. So habitability model like this help to interpret and add context to any biosignature detection. So why habitability models? First, to determine the relevance for life of one or more environmental variables. For example, a challenging problem, let's say you want to quantify the importance for life of magnetic field, of the magnetic field of a planet. So you use this model to see what will be the net effect on life of this variable of interest. So that's one application. To select a target for observations, follow the habitability, to compare results from observations of models. For example, determine the global habitability of a general circulation model. Let's say you are doing many models, 1D or 3D models, and you are getting, and you have a lot of information about the planet because these are pure simulations. Then we draw this model, biosphere model or biomodels. Then you can decide, or from all the simulation, what are those that can be considered more habitable. In this case, it means that they have a more mass and energy available. They can produce more mass and energy available for life in the simulation. Why these models? Also to provide context for biosignature detection. Habitability is possibly correlated with the abundance and diversity of life on Earth, but we don't know if this is true elsewhere. Finally, to understand ecosystem on Earth and their potential elsewhere, from the solar system to exoplanets. And certainly, habitability models are not here to determine if planets are habitable or not in the common sense. Otherwise, why biologists are doing this? We wouldn't be using them on Earth because we already know it's habitable. So what's the point? It's because all the first five steps are necessary. That's why these models are necessary. In conclusion, habitability is the suitability of an environment for life. Habitability is proportional to carrying capacity, and at least in the traditional models of the ecologies, or the available mass and energy for life in our model, which is just an extension of this idea. Habitability models help to quantify and integrate the effects of one or more planetary and stellar variables on life. A library of habitability models for different variables, species, etc., is usually created to study a system. It's not that if you want to study a system, you are asking for a single model with all the variables. No, that's not the best approach. You just start to divide a problem by parts, considering just the atmosphere, just the water, and different set of variables. And so all these models will provide an idea of what is going on or what you might expect in the system. And habitability modeling is in astrobiology is an emerging field. I can say the same thing for these models in ecology, but just do this exercise. Try to look in any biology textbook or ecology textbook for the word habitability. But then try to look for habitat and habitat suitability and papers on habitat, and you will be overwhelmed by how many science have been done. So I just scratched the surface of how these models are applied in ecology and how they can be integrated and extended into astrobiology field. And so we have a lot of work to do, and that's why I say this is an emerging or re-emerging field. So you can check our paper that will be available soon, probably May next month in astrobiology. But we also have now in the archives a white paper that we submitted to the Planetary Science and Astrobiology Decadal Surveys, which includes these ideas and in this case applied for planetary science. In our paper, we apply to the astrobiology. We're more focusing on the astrobiology. So thank you very much, and I am open to questions.
All right, thank you, Abel. I'll clap on behalf of everyone. Yeah, throw those clapping emojis up. And if you have a question, you have two options. You can raise your hand using the reactions button on your Zoom window, and I'll call on you to unmute yourself and ask your question of Abel, or you can put your question in the chat. So one of two options. David Catling, you are up.
Hi, Abel. Thanks for your talk. It was very interesting. So I'm wondering how useful your sort of empirical parameterization is for surface temperature, because you had surface temperature in one of your slides was 1.1 times the equilibrium temperature. But that's based on, you know, a small number of real planets, which is Earth, Venus, and, well, actually not Venus, because that's just for low pressure atmospheres less than 10 bars. So really the two real planets you have that feed into that are Earth and Mars. So really there's two real planets and then some modeling planets. And then you have to exclude things like Venus, because Venus is not 1.1 times the equilibrium temperature. It's a bigger multiplier. So, and also you have a 50% error bar even with this. So isn't there a big uncertainty, because you might have different types of clouds, you might have more exotic atmospheres that we're not modeling, or we know nothing about yet because we haven't discovered them. So how useful is this when even with your model you have a 50% uncertainty, but really there's probably a bigger uncertainty?
Yes, that's very true, David. And the thing is, as I showing here in the, let me put the rapid system. So here is the same model with that large uncertainty. At least we can constrain that assuming 10 bars. If I use here 1 bar atmosphere, I can constrain further, probably 25%. But it is true that this model is based mostly on simulation, 1D and 3D. And so far, what we have seen here is that even putting different atmosphere conditions, at least for the 1 bar case, that there's many simulations, different conditions, the rotational planet, that doesn't change much. I think that the most important thing here is that these equations simplify, at least, because this kappa factor does not depend on the stellar flux. So no matter if the planet is close to the star or farther away, actually, if it is farther away, this decrease the actual change in temperature, and it will be more easy to predict the temperature. But it's more uncertain when it's getting closer with respect to the flux. And at least it can constrain you some temperature. What I am mostly interested is how this kappa will change with pressure. And as you see here, we have a few sample points from a simulation that got up to high pressure, up to 100 bars of CO2. But I would love more, and I'm trying to collect from the community simulations beyond that, what we have, what we can do, simulations of general GCMs. So we can also try to define this relation about the kappa and pressure, which we know is strong.
Okay, thanks. I guess my comment would be that it depends on how well those 3D GCMs are modeling albedo, which is a big factor. But for the Earth and Mars, it's actually not too different. It's about 0.3, but maybe there could be big differences in albedo, and maybe that will that could be a confounding factor. But yeah.
Yeah, I have to rely in those simulations. So,
yeah.
But at least they're taking as many different simulations as possible, and a pattern is emerging. So, is it right, or everybody is wrong?
All right, thanks.
Okay, thanks.
Yeah, great question and excellent answer. Seeing no other questions in the chat or raised hands, I'll ask one of my own. So Abel, yeah, it was very interesting to see that the deep sea environment was so low on your habitability index, especially given that we know of ecosystems at hydrothermal vents that have biomass densities that rival rainforests. So I was wondering if you could speak to potential exceptions to your quantification metrics when you consider local scales, or if these local scales are sort of averaged out when you take a planetary scale perspective, and that's pretty much what you're doing here.
And so, yes, okay. And these are averaged out. One of the things when you're trying to apply this model, you have to decide a region of interest. And I mean a region of interest in terms of space. So it will be quite different if you a hydrothermal system, that then you have to go to the local scale. And then also on time, because there's some dynamics there. So you have to pick that, and so your model, starting by the same, the good thing here is that starting by the same mathematical framework, then you go to a particular case, and your solution is only good for this particular case. And you know what ecologists do? Create libraries, full libraries of, okay, habitability, suitability for this species in this kind of environment, same species in other kind of environment. So it's a zoo of metrics depending on the scale, condition, and species, and that will be the goal. But at least you have a standard And when you have this standard, and because you are always compared to some environment, so the scale, you can normalize the scale 0 to 1. And when you see a 1, if I'm telling you that this environment is 1, is habitable in a common sense. No, it just means that it's better compared to some standard. And 0 is not compared to some standard. And that will be common no matter what type of environment you use. But it is a relative scale depending on the scale. So that's why I presented one for Mars that uses relative humidity and temperature, one for exoplanets that use large properties of the planet. And it was focused in the whole biosphere. So that's part of the problem, and something that we learn from ecologists, that you have to agree at the beginning of everything to the scale that you want your model to be applicable.
All right, excellent. And I will ask Don Brownlee now to join us. He's been waving his physical hand and I missed him. Sorry about that, Don.
Oh, that's okay. Thank you for the talk. I was just wondering what you thought about habitability of subsurface microorganisms, so-called slime habitats. Some people think a significant biosphere on Earth is subsurface. It may be almost impossible to detect from a distance, but it's also conceivable that's the most common kind of life in the universe, because if these organisms live oxidizing Fe2+ to Fe3+, and do it slowly with low turnover rates, they can do it for billions and billions of years, even around dim stars. And I don't know, it's kind of an open question, but it's commonly overlooked, but it may be the most common life in the universe.
So probably for that kind of life, there might not be a response curve. So we can, at least if there's an experimental laboratory, usually these response curves are measured in the lab, are determined in the lab, or they can be estimated by testing different range of operation, of growth or activity of the life of interest. But at least we can, because we need the fluxes of mass and energy, I think that the harder problem to solve that, if you're considering it's a surface environment and you assign a particular volume just to calculate all these fluxes that goes in and out of your volume of interest, that's a hard problem. So I don't think that that problem is only complicated because of habitability, also because in the terms of physics and chemistry and actual fluxes, that's a complicated problem. Once we solve that, then we can start telling and asking about the habitability of the system.
But if they're truly living by chemical energy, they really don't need any extra energy. If they're just oxidizing Fe2+ to Fe3+, and we do it at a low rate, I don't know. It's kind of frustrating to think about, but it's possible.
I like those.
The advantage is that in the solar system, any place that's not too hot, it's a potential habitat for these things.
I like those questions because those are challenges, and our plan now is to apply this model to as many cases as possible, as with people that are asking with help with these models. And one part is the mass and energy fluxes, but state variables like temperature, relative humidity are related to the Q factor here. And that's something also hard, but those are good examples that we need to work out. At least simple variable first, and that models, and then go further and further, adding more complication.